morning, good afternoon, good evening to everybody who's participating in today's webinar. So we're going to talk about pharmacokinetics and really address the question whether this is a step towards precision medicine. So I'd like to begin with the mission of the National Hemophilia Foundation, which focuses on enabling people and families to thrive. And that is our ultimate goal. And we'll see whether pharmacokinetics can help us enable people and families to thrive. So what will I tell you in today's presentation? We'll start with some basics. We'll talk about the standard of care for hemophilia treatment in the developed world. And then we'll talk about health equity, which in my opinion, I'm giving you the conclusion here, remains to be achieved. So let's begin with some basics. So we'll start out with what is precision medicine? So precision medicine operates on the principle that most medical treatments are designed for the average typical patient, a one-size-fits-all approach. While precision medicine, sometimes known as personalized medicine, is an innovative approach to tailoring disease prevention and treatment to an individual. It takes into account differences in people's genes, environment, and their lifestyles, and it targets the right treatment to the right patient at the right time. So that's the fundamentals of what precision medicine is. Now, when we think about that, we can think about a couple different types, and I borrowed the next couple of slides from Dr. Peter Marks at the Food and Drug Administration. So it's about creating the right drug for the right patient at the right time. So Dr. Marks told us about customized and created products. Customized products are for the same indication and the same mode of action. So for example, there's different ways to treat, for example, pancreatic cancer, or in our case, different ways to treat hemophilia with drugs versus created products, which are for different indications and may have different modes of therapy. And these might be gene therapies for two different types of hemoglobin mutations using the same vector backbone, or in our case, maybe treating hemophilia A and B with the same vector, but inserting a different gene into that vector. We also have these notions here about what is ready to wear, and that's similar to buying a suit off the rack. You put it on and it fits perfectly. Where made to measure is something where you buy a suit or a dress and it's tailored to fit you according to your measurements. And then of course there's bespoke, which is really creating the entire product from start to finish, designed and tailored to specifically fit you. So this is what we're really talking about, is the made to measure and the bespoke types of products, which is really where we really get into what precision medicine is all about, personalized medicine for that individual patient. So let's ask the question, what do people who live with hemophilia want? Now, there's been a lot of research to try to understand this. And I think some of the research points us in the direction that people want to be free from bleeding and the burden of the treatment to prevent that bleeding. They also want to be more active. And of course, they want to lessen the physical and mental impact of hemophilia on their daily lives. So let's begin now and think about this. But the ultimate bottom line is they really want to be free from, as much as possible from the biological, the physical, and the mental grasp of hemophilia. Living a hemophilia-free life is really what the ultimate desire is. So let's begin with talking about free from bleeding and the burden of treatment. So we look at the WFH guidelines for the management of hemophilia, and I point you to chapter 6 on prophylaxis in hemophilia. So the first question we ask is, what is the definition of prophylaxis? Well, in the past, this was regular intravenous injections of the missing clotting factor, either factor 8 or factor 9, for people with hemophilia A or B, respectively. And the goal was to increase the level of the missing clotting factor sufficiently to prevent bleeding. The new definition is the regular administration of a hemostatic agent or agents with the goal of preventing bleeding in people with hemophilia while allowing them to lead active lives and achieving quality of life comparable to the non-hemophilic individuals. So you can see that the definition of prophylaxis has evolved, especially as we have now non-intravenous or subcutaneous products that can prevent bleeding. So what are the classes of drugs to use for prophylaxis? We have plasma-derived, standard half-life, extended half-life, and emicizumab, a bispecific monoclonal antibody. All of these drugs are thought to be safe and effective. While the first three, plasma-derived, standard half-life, and extended half-life, require intravenous injections. So I'm going to focus for the beginning part of this on these three classes of drugs for prophylaxis. So I want you to understand that currently, in using any of these types of drugs, pharmacokinetics is typically utilized to optimize the dosing for people with hemophilia. Pharmacokinetics, there's a few terms that you need to understand. Absorption, that's how the drug is taken into the body. Distribution, how it moves around the body. Metabolism, how it's broken down. And elimination, how it's eliminated or your body gets rid of the drug. So pharmacokinetics is like a fingerprint. Every PK profile for an individual is unique, and nobody else's PK profile is quite the same as yours might be. The value in using a PK profile is your doctor can take a blood sample, and after one or more infusions of factor VIII or factor IX, then the amount of factor VIII or factor IX in the blood can be measured in the laboratory. The doctor can generate a unique PK profile based on those measurements, and then you can determine the best treatment plan, the right amount of factor to infuse at the right time with the right dose and frequency. So this is the basics of pharmacokinetics. When we look at pharmacokinetic measurements, there's a few terms you need to understand. Peak. That's the highest level that you achieve after the injection. We also have something called area under the curve. This is the amount of factor or the total amount of factor that's in your body for the duration of time. You also have a half-life. Half-life is defined as how much is eliminated in a specified amount of time. How long does it take to get to half of what you started with? So if you inject it to a peak of 100%, how long does it take to get to 50%? There's also clearance, and clearance is the amount of factor which is eliminated from your body over time. And then lastly, trough level. How low does it go? So let's look at these terms in a little bit more specific terms. Peak is the maximum level, and that's akin to the amount of battery life that your cell phone may have when it's fully charged. We would hope it would be at 100%. Area under the curve is the amount of charge that is used by your phone over time after you fully charge it. So that's how long you have and how much charge is utilized. Half-life is similar to how long does it take your phone to go from 100 to 50%. Clearance is how quickly your cell phone battery discharges. And then trough level is how low do you need to get until your battery notification appears that you need to recharge. So these are some simple ways to think about pharmacokinetics. So what's the outcome with standard prophylaxis? And here you can see three different studies from 2002, 2011, and another from 2007 that look at the first two with intermediate dose prophylaxis, and the last one, the Manko-Johnson article, with full dose prophylaxis. And you can see that prophylaxis in the blue reduces the number of joint bleeds in both children and adults, the overall number of bleeds in children in the middle set, and then overall bleeds in children as well in the Manko-Johnson study. And you can see that the reduction is between about 75% and 95% with standard half-life factor VIII replacement products. Similar reductions are evident in studies with hemophilia B and factor IX prophylaxis. So prophylaxis is good, but patients need more and want more. They want less infusions. They would like to have higher trough levels to prevent bleeding and to be more active. And of course, they would like to see non-IV administered products. Well, extended half-life products can achieve less infusions, and they can also generate higher factor trough levels, achieving two of those three goals. But of course, extended half-life products still need to be administered intravenously. And these slides come from the World Federation of Hemophilia treatment guidelines that I showed you previously. So people also want to be more active. Can we explore that a little bit more? What's the data about being more active for people with hemophilia? So there's a nice study that I wanted to review with you, and that's called the HemActive study. So it asks the question, what is the impact of extended half-life versus standard half-life treatments on physical activity, treatment satisfaction, and adherence for people? So in this study, there were 209 participants with moderate or severe hemophilia A. 3/4 of those individuals had severe hemophilia A. The age ranged from 2 to 65 years, with about a third of the individuals under the age of 18, for which the caregivers participated and responded to the survey. The survey was a 25-minute IRB-approved web-based questionnaire that included a 5-level scale to collect activity levels. People to participate in this study were recruited from Europe and the United States from March to May of 2018. Now, it's important for us to remember that date, 2018, and that'll come back in a few minutes. The findings of this study showed that 82% of the participants were on prophylaxis. The distribution of standard and extended half-life products is shown, about half and half on each of the products. 11% were using plasma-derived product, and 2% were unknown. And this data was presented in abstract form in 2019 at the International Society on Thrombosis and Hemostasis Congress. There were several insights that came from this study, and this slide depicts 5 of these the participants' goals and desires. One is that hemophilia patients adjust their activity levels due to both experience and fear. They know what in the past has resulted in bleeding, and they hope not to bleed again. They also are fearful of future bleeding. The second insight was hemophilia patients want to be more active and believe less pain and better protection would lead to their ability to be more active. The third is that patients believe their condition has made them a stronger person. The fourth insight is that some patients personalize treatment as they prioritize activities over medication adherence. And what this may mean is that people will dose based on their level of activity or their anticipated activity levels in the coming hours or days. The fifth insight is that patients describe wanting a normal life that allows for flexibility and freedom, though some have accepted and settled into their current lifestyle. And this is what we now consider the disability paradox. So we know that patients want to lead a normal life. They want to be active, but how do we help them achieve this level of activity through education, advocacy, and of course, through better research? So let's look at the next thing that we said that people may want: lessen the physical and mental impact of hemophilia on their daily lives. So I'm going to turn to another study here, the PROBE study, to understand how well we're doing. And the PROBE study, or the Patient Reported Outcomes, Burdens, and Experience Project, developed an evaluation questionnaire to assess patient-reported outcomes in people with hemophilia. Part of this, what they're looking to collect in terms of outcomes, is shown on the table to the right. There's a desire for reduced burden of living with hemophilia and their impacts on life and family and education and employment and other activities. In addition, there are parameters to look at the complications associated with hemophilia and its treatment. For example, joint status, pain and depression and anxiety. Also, obesity and other comorbidities are evaluated in this PROBE questionnaire. So I want to now turn for a second to this, on this, to understand how well we're doing before we turn to the next slide. We'll come back to this in a few minutes and we'll look at the data from this study. But I think the bottom line here is that we're not doing as well as we potentially could. There's still significant burdens of disease that people experience, as well as complications. So when we look at the standard of care for people with hemophilia in the developed world, what is the impact of these treatments on their survival? And this is a study that was just recently published from a group in the Netherlands that looked at mortality and life expectancy and causes of death of persons with hemophilia between 2001 and 2018. So again, an important benchmark for us to consider, 2018. And in this study, you can see that there were more than 1,000 individuals who were recruited to participate. Their mean age is evident there, and you can see that it's about 34 years, plus or minus 20. So we have people ranging from about 10 all the way to 50 or 55. The severity of hemophilia, about half of them have severe disease. The remainder mostly have mild disease, and then there's a few people with moderate. And you can see, as expected, about 9 out of 10 individuals have hemophilia A, and the remainder have hemophilia B. So given that population, what's the impact on their survival? Trying to get my slide to change. So on here, you can see the lifetime survival curve in the different cohorts: severe to the left, moderate in the middle, and mild hemophilia to the right. And this, again, are cohorts between 2001 and 2018. And what we can see is that the severity of hemophilia still plays a significant role in their overall survival. And now in the next slide, we'll compare that to healthy people in the general population. So here we see the median life expectancy in patients with hemophilia in the Netherlands between 1973 and 2018. You can see the 4 different cohorts shown on the bottom. Cohort 1 is the earliest, and cohort 4, far to the right, is the 2001 to 2018 cohort that we saw in the previous slide. At the top line, you can see that's the general population. So that's the median life expectancy over those periods of time for healthy individuals in the Netherlands. You can see the solid line in the middle is for all hemophilia patients. But again, we see that people with severe hemophilia have a significantly decreased median life expectancy compared to moderately severe and mild hemophilia patients, and definitely different than the general population in the Netherlands. So our treatments are good, as I said, but they're not sufficient to normalize lifestyle. The conclusion here of this paper was survival in patients with hemophilia in the Netherlands has improved over time, but is still lower than the general population. It tells me that we have more to do to improve the overall outcome for people with hemophilia. So here's a probe score for individuals now. This was presented in 2019. This looked at 366 participants whose mean age was about 45 years, and they compared people with non-severe hemophilia to normal controls. And what you can see here is even people with non-severe hemophilia had increased burden of disease and a lower outcome compared to the healthy controls. So again, as we saw previously, their life expectancy is decreased with hemophilia compared to healthy controls. And the more severe the disease, the lower the survival rates are. And as well, in terms of burden of disease and outcome and their experience, demonstrated by the probe score, people with hemophilia are doing worse than the healthy controls. So all in all, we've still got a lot of work to do to improve the quality of life and the life expectancy for people with hemophilia. So I asked the question, do we have a better mousetrap? And I point to the approval of Emicizumab for the treatment of hemophilia A in patients with and without inhibitors. This occurred on October 4th, 2018. There's that important date to look at. So all of the data that we've talked about previously is in the pre-Emicizumab era. We'll now be able to evaluate what the impact of emicizumab is on some of these outcomes. For example, on life expectancy, on the occurrence of joint disease, on the occurrence of depression, and all of those uh, parameters that are measured in a probe score. So let's now turn our attention from the first three categories of drugs to emicizumab, which I said is safe and effective and is administered by a subcutaneous route, decreasing the burden of administration just by virtue of its route of administration. So the benefits of emicizumab include less infusions, potentially none if there's no breakthrough bleeding, higher factor trough levels. Well, it's not really factor levels, but it's the effective hemostatic level is potentially much higher than it could be achieved with factor replacement therapy. And of course, it's not administered through a non-IV route of delivery. So what about bleeding? So let's look at breakthrough bleeding and perioperative bleeding in patients who are receiving emicizumab. So the first study that I wanted to talk about was one that was just recently published by Cohen and colleagues in Pediatric Blood and Cancer just a couple of weeks ago. And this was a retrospective review of 28 children with hemophilia A, and they all had severe disease defined as less than or equal to 2% factor VIII activity. 13 of the 28 had a history of an inhibitor, and this analysis was done at Texas Children's Hospital from 2018 to 2021. So all of these were individuals who were switched from factor replacement therapy to emicizumab during that time period. The mean annualized bleeding rate decreased from 3.61 with factor replacement therapy to 0.44 with emicizumab. So that tells us that people that are treated with emicizumab have a tremendous reduction in their annualized bleeding rate, but not quite to zero. So these individuals are still bleeding about once every other year. There was also another review that I wanted to point to, which was done by Warren and colleagues at the University of Colorado, and also reported just a few weeks ago in Research Practice in Thrombosis and Hemostasis. They conducted a retrospective review and telephone interview of 68 children and adults who were switched from factor replacement therapy to emicizumab prophylaxis. There were 68 individuals who participated in this retrospective review, and of those 68, 25 experienced no bleeding. So they had 0 bleeds. But we can see that the vast majority of individuals did report bleeding after they switched to emicizumab. And in fact, 5 of the 68 experienced more than 8 bleeds per year. We'll look at this in a little bit more detail on the next slide. We don't quite have enough experience yet understanding the benefits of emicizumab in perioperative bleeding. There have been a number of short case series reported, as well as some clinical trial data on the use of emicizumab to prevent bleeding during and after surgery. But I think that we need more robust data, and importantly, we need real world data like the studies of Cohen and Warren and colleagues to really demonstrate the benefit of emicizumab in the real world, both to prevent breakthrough bleeding as well as perioperative bleeding. So this is data from the Warren paper that looks at emicizumab initiation and bleeding outcomes in people with hemophilia. In the left-hand side, you can see the bleeding pattern after starting emicizumab. And what we can see here is that over time, the number of bleeding events definitely decreases. And you can see that as a pretty prominent trend. As time goes on, the number of bleeding events decreases both for spontaneous as well as traumatic bleeding events. But it's important to recognize that there's still individuals who are bleeding even months after they started on emicizumab. The location and nature of bleeding events is shown in the right-hand figure. And what we can see here is that there are individuals who continue to have bleeding, either spontaneous or traumatic, despite the emicizumab. So again, I think it's around optimizing the treatment. This is still a very young drug for us to know how to use and optimize its treatment. So I think, again, more real world evidence is needed to really support the optimal use of emicizumab. And how can we tailor not only the use of factor replacement therapies, but also emicizumab to the patient. So I think we're still learning how to optimize uh, all of these drugs to enhance the protection against bleeding for people with hemophilia. So I asked the question, is gene therapy the answer? There's been a lot of talk about gene therapy. So let's investigate this a little bit. So here's a paper that Dr. Steve Pipe, who chairs our Medical and Scientific Advisory Council, wrote in 2020 around the delivering on the promise of gene therapy for hemophilia. It's a single treatment that is designed to provide continuous factor expression and essentially achieve a functional cure. That includes freedom from bleeding. We're hoping to have normalized hemostasis, which eliminates the need for continuous factor replacement and hopefully will result in optimized health and well-being. So the first thing is the promise of gene therapy. We believe that these are safe, but with that jury is still out. We still need more data on the overall safety of gene therapy products, especially AAV-based gene therapies. There's a number of questions that continue to be a top of mind regarding the safety of gene therapy, including the hepatotoxicity, the elevated liver enzymes that result after the dosing with an AAV-based gene therapy, and how to control that liver toxicity, the potential for hepatocellular carcinoma, and even other malignancies due to integration of the vector into the host genome. And of course, there's a number of other unknown risks that are yet to be fully understood or appreciated. In terms of efficacy, we can measure things like the frequency of bleeding, the factor activity level, the duration of expression of the factor, the impact on chronic pain, healthcare resource utilization, and mental health. And I point you to this paper by Iorio and colleagues from 2018 that described a core outcome set for gene therapy in hemophilia, which was called the Core Heme project. And I think it's really important that we recognize we have a defined set of both safety and efficacy parameters that should be used to measure the value and the efficacy as well as safety of gene therapy products in clinical development. So what can we learn about gene therapy, and are we delivering on the promise? Well, I would say, are we meeting the expectations of a cure in terms of our current variability in response? We have some patients who respond quite well. We have other patients who don't respond terribly well. So the variability in patient response remains a concern. The predictability of that response. Am I going to be able to get 100% factor activity? Am I going to get 1% or no percent activity? Or am I going to be somewhere in the middle? We're not quite able to predict what that factor level is likely to be for an individual patient. Durability. We know that this is a question, especially in hemophilia A, where we've seen in the longest running trial out to now 5 years, that the factor level, the factor VIII level, continues to decrease over time. Whether that's the case for hemophilia B remains to be determined, but at least at this point, we have a couple data points to look at. One is the long-term data with hemophilia B that is now over 10 years, where we have low but consistent factor levels in that 2 to 3% factor IX activity range. So the durability in factor IX seems to be somewhat better, but at a lower level. And then we also have data from a couple other trials that indicate durability to about 2 to 3 years at maybe higher levels because of different vector designs. So in terms of durability, there may be some differences between hemophilia A and hemophilia B. So we have to look over time to see what that durability is really likely to be. And of course, there's questions about access to care and the affordability of gene therapy. So I'll point you to a couple papers that were written. The first one, as I said, was Dr. Pipe's paper on delivering on the promise of gene therapy. But I think a really important paper is by Dr. Pierce, who wrote about our gene therapy for hemophilia. Is it meeting the expectations? Are they really matching up to reality? And I think it's clear here that we need more data on all of these areas: the variability, the predictability, the durability of our gene therapy products for both hemophilia A and B, as well as the accessibility and the affordability of these products over time. So I'll say that for gene therapy, the jury remains out. And this was evidenced by a recent Food and Drug Administration, the FDA, Center for Biologics Evaluation and Research, conducted an advisory committee meeting on September 2nd and 3rd. And the express goal of this committee meeting was to discuss the toxicity risks of AAV vector-based gene therapy products. And the discussion topics included the cancer risk, that oncogenicity risk due to vector integration into the host genome, and other safety issues that were identified in preclinical and clinical evaluations of gene therapy products. So there's more to come from this meeting, and we'll be having a summary of this meeting in the next couple of months for the community. But I think it's still early in the process to fully understand what the safety as well as the efficacy of gene therapy for hemophilia is. It also doesn't talk much about the accessibility of gene therapy and how we're going to be able to access this innovative treatment throughout the community. And more importantly, who's going to pay for this and how is reimbursement going to be handled? So we've got a lot to solve still for gene therapy. So given all of this information, how is anybody going to make a choice? So let's think about how we choose and undertake a decision-making process. You have two red cars there. How do you choose which one you'd like to purchase? You may go for comfort and color and shape and performance and the access to the vehicle. You may opt for more safety, more predictability, more durability, more effectiveness, or you may consider the cost. So I think it's challenging, and we cannot make a decision easily for any of these therapies, whether it's a plasma-derived, a standard half-life, extended half-life product, subcutaneous therapy, or in the future, gene therapy or other innovative novel therapies that are likely coming our way in the bleeding disorders community. So we need to have some way to consider how we're going to make these decisions moving forward. So the first thing I'll point to is, as we started out this discussion, each person is an individual. What's right for one individual may not be for the other. So we have to consider each person as an individual, and we really think about that precision medicine approach. Do we want to have designer drugs for the treatment of hemophilia, or for that matter, for any blood or bleeding disorder? So let's consider the individual first. And we have to think about what matters to the individual patients. So this is some really nice work by a group that looked at patient-relevant health outcomes for hemophilia care. And the goal was to develop some international standard outcome sets. And what we see here is, in the first group, health status that's achieved or retained. So we talked about survival, and we talked about that in terms of the data from the Netherlands, where we know that survival in the hemophilia community is not equivalent to their unaffected peers. So what impacts survival? Of course, cure or lack thereof, and the impact of the disease on its life expectancy, the degree of health or recovery, and that is related to the ability to engage in normal daily activities free from bleeding episodes. So the first thing is, okay, an individual patient has to evaluate their health status and how they're going to factor that into their equation. The next level is the process of recovery. And its time to recovery could be measured in the number of days lost from school or work, or the disability or disutility of care or treatment. And that may be related to chronic pain or other complications of the disease. And again, an individual has to consider each of these for themselves and how much value they put into these parameters. And the last level is around sustainability of health. And it's sustainability of health or recovery and the nature of recurrences. So we'd like to sustain physical functioning. We'd like to be able to do the things that we would want to do in everyday life. Are we able, for example, to walk up and down stairs, to participate in activities with children or other family members? Are we able to go on a long hike? Can I walk 30 minutes to my doctor's appointment? Or can I ride a bike? Or can I even perform my daily activities at work? There's also long-term consequences of therapy in terms of social functioning and mental health. And one of the really important, some really important work that's going on right now is in the Core Heme project, where we're really trying to develop mental health outcomes and a scale to evaluate mental health in terms of these innovative therapies, in particular gene therapy. And then, of course, social functioning. Are we able to participate in life the way that we would like to? So what matters to the individual patient may be very different, but we need to take all of these things into consideration. So again, how to choose a treatment for hemophilia. This is a complicated process, and there's many different factors that go into this. So I'd like to propose a way that we can think about this, and that's through what's called shared decision making. So shared decision making is a stepwise but fluid process that includes discussion of goals, values, and desires on the part of that individual with the disease, focusing on what matters to patients and their families. This results in treatment decisions arriving from a balanced consideration of risks, benefits, and alternatives between available management options and patient preferences. So the healthcare professional and the individual who lives with the disease participate jointly in the decision-making process. Now, this is a very different approach than what has historically happened in medicine. We've had a very paternalistic, maternalistic medical system where a physician or a healthcare professional guides the patient and makes the decision for that individual about what the best treatment option is. And then it's incumbent upon the patient to adhere to that decision that's not been made by themselves. It's been made by their healthcare professional. In a shared decision-making approach, it's a joint effort. It works together to enhance the decision-making process and really puts the patient in charge, because who can be more of an expert than somebody who lives with their disease? So the share or shared decision-making approach is to seek input from the patient's participation. Now, again, this was written at a time when we were still focused on evolving from this maternalistic, paternalistic approach to decision making. So really it can come from either the patient or the healthcare professional to involve the other in this process. And I would encourage all people who are listening to this, if you're a healthcare professional, to engage with your patients in shared decision making. And for those patients who are listening to this, to engage your healthcare professional in a shared decision making process. The second step is to understand and explore and compare treatment options. So that's getting as much information as you possibly can about what all the options are. And then it's assessing the values and preferences. So it's a comparison. It's matching up the information that you have in step 2 to what your goals and desires are in step 3. It's reaching a decision mutually and in collaboration between the healthcare professional and the individual who lives with the disease, and then evaluate the decision afterwards. So we can go into this in a little bit more detail. So I want to thank Declan Noon, who provided the next series of slides to me. And there's a number of different areas that we can be thinking about, especially when considering goals and expectations of any treatment. There's some that are more important to patients and others that are more important to healthcare professionals. So the first question is, in terms of goals and expectations, meeting the patient where they are. This is an important component. It's understanding the goals, the lifestyle, the motivations that patients identify, what life stage they're at. So it's really understanding what's important to the patient and the family member. The next is there's a number of what are called lagging indicators. These confirm but do not predict long-term trends. They're not additive, and they're used to look at the impact on a group, but they're difficult to apply to an individual. So what do we mean by that? These are things that we're commonly measuring: quality of life, mobility, activities of daily living, measures of pain and comorbidity. So these are what are termed lagging indicators that are good for populations, but maybe not so good for an individual. And then lastly, we've got outcomes that are important to healthcare professionals, but not really terribly relevant necessarily to the patient. These are necessary, but are not sufficient. They're easy to measure, and that's why we've measured them historically for a long time. But the impact for patients is minimal, and it's mostly relevant to clinicians and importantly to payers. These are things like the annualized bleeding rate that we've talked about so frequently, treatment administration, the factor expression level that's achieved, whether that's from a pharmacokinetic assessment or from the use of a non-factor therapy or from gene therapy. And of course, treatment consumption. How many international units per kilogram are you using on an annual basis, as well as safety issues? So all of these apply differently and have to be valued by both the healthcare professional as well as by the patient who's making these decisions. So I want to turn to the last part of my presentation in the last 10 minutes or so and talk about the bottom line here is where I think health equity remains to be achieved. So what is the ultimate goal? And this is a slide that I took from a paper that was published by Mark Skinner and Diane Nugent and others that looked at achieving the unimaginable, health equity in hemophilia. And they state in the paper that the real goal for treatment should be a functional cure and achieving health equity. And what the figure on the right-hand side looks at the level of protection along the bottom, and then clinical outcomes and factor levels that are achieved. And what we can see here is that there's a minimum amount of protection that's necessary for survival. We looked at that data from the Netherlands, and we saw that there was significant improvements in the survival of individuals, but it doesn't take very much protection. We don't have to go very far from the left side to the right to achieve survival. Well, what about minimal joint impairment? We know from the joint outcome study that I showed you previously, as well as the other two studies, we can prevent joint bleeding and hence prevent joint damage. But it doesn't, again, take very much to be able to do that. And you can see that by achieving levels of somewhere between 1 to 3% factor 8 or factor 9 activity, we may actually be able to prevent from much of the joint disease. But we may not be able to prevent all of it because of the occurrence of subclinical bleeding that may occur at somewhat higher levels of factor activity, especially with more strenuous physical activity on the part of the patient. The next level is the ability to engage. What are we able to do and participate in daily activities, but of course with the freedom from bleeding and other complications? This takes a little bit more level of protection. We have to move up the scale and move further to the right. And then we've got the ability to achieve normal mobility. So this would be freedom from joint complications. And as we move further up the ladder and really move towards the top, it's achieving normal hemostasis, which is where we would then be able to say, well, we're at an equitable time and somewhat equivalent to somebody who does not have hemophilia. And it's really important that we think about both the health equity as well as the functional outcomes and eventually the functional cure for patients. So I really like this model that's been developed that helps us understand the levels of protection that we've moved through historically and are currently on that journey towards health equity and normal hemostasis. But I believe that new treatments and new treatment paradigms are needed that are based on the defined needs of people with hemophilia to really be able to achieve health equity. And I would argue that this is not just for hemophilia, but it's for other bleeding disorders, including von Willebrand disease, the rare and ultra rare bleeding disorders, platelet disorders, and of course, for all blood disorders, that we should really be striving for health equity in blood disorders. And again, new treatment paradigms are needed based on the defined needs of people with blood disorders. And that includes people with, for example, sickle cell disease, thalassemia, and other blood disorders, as I said, including von Willebrand disease and platelet disorders. We should be leaving no disorder behind. And we're really looking towards our future therapies to be able to deliver on a functional cure and achieving health equity for people with blood disorders. So as you know, the vision for the National Hemophilia Foundation is a world without inheritable blood disorders. How are we ever going to be able to achieve that vision? Well, I think one of the first steps is addressing the social determinants of health. Social determinants of health are conditions in the environment where people are born, where they live, where they learn, where they work or play, worship, and age that affect a wide range of health, functioning, and quality of life outcomes and the risks that are associated with this. So the Department of Health and Human Services has an initiative around something called Healthy People 2030. And these are decade-long plans to address specific health needs to improve the life and well-being of people in the United States. I think there's a lot of work that the National Hemophilia Foundation, its chapters, and hemophilia treatment centers and the rest of the community can do to address the social determinants of health. That includes education and access and quality of care, economic stability, social and community understanding of these social determinants of health, looking at the neighborhoods where our patients live and the environment that they exist in, and of course, access to healthcare and quality healthcare at that. So what are the challenges that I think we're dealing with? Obviously, the coronavirus pandemic continues to rage, not only in the United States, but around the world. But I think another challenge for us is diversity, equity, and inclusion. And we can all talk about equity, but it's really what I'm hoping we can do is move from equity, where we give people what they need, to liberation, where it's really removing the barriers that are present. And by doing that, we can only do this through inclusion. And inclusion includes bringing traditionally excluded individuals and or groups into the process and activities and decision making and policy making, which is so important. People need to participate in ways that share power and ensure equal access to opportunities and resources. So I think we'll be able to achieve health equity and, importantly, bring that promise of precision medicine forward through inclusion. We've now started thinking about our experts who live with these diseases as subject matter experts. It's the inclusion of subject matter experts, people with lived diseases, whether that be von Willebrand disease or a platelet disorder or hemophilia, into the discussion and into the decision-making process so that we can recognize and ensure that their voice is heard and that their opinions are respected and represented. So how do we achieve inclusion? It's by creating that community. And that's one of the things that I'm looking for all of you to be able to help us with. It's creating that community of collaboration. It's really creating the community of everybody working together. And as I said in my first address to this community, now 18 months ago, we're stronger together. We have to bond together as a community. And I think the subject matter expert, the person who has the lived experience of the disease, sits at the center of that. And this slide is courtesy of one of our subject matter experts, Esmeralda Vasquez, who participated in the research state of the science and is a member of our steering committee. And what Esmeralda taught us was that a subject matter expert sits in the middle and needs to be able to self-advocate and then encourage others in her or his similar position to be self-advocates, to participate in research, to ensure that their data is part of the concerted data effort to move clinical data into research and then research data back to clinical practice, to be a member in advocacy and to speak their voice and participate in national and local advocacy efforts, to both provide as well as receive education. And this is so important. We can learn so much from our subject matter experts by giving them a seat at the table and letting their voice be shared with others. As well as the last area that I'll point out is individual healthcare decisions. We talked about shared decision making. The subject matter experts need to be at the table with their healthcare professionals, making and encouraging decisions that work for them. So how do we make all of this happen? It's by participating. We all need to be participants in this process, because only through this will we really be able to realize the impact of all of the things that we've talked about today. So with that, I'd like to thank you. And to answer the question, is pharmacokinetics really a step towards precision medicine? I think it is, but I think we've still got a long way to go to really be able to realize the goals that we've set forward today and achieving health equity. So with that, I'd like to offer the opportunity for a couple of questions.
Thanks so much, Len. We appreciate that. Because the horrible echo, I sent the question via the chat.
All right, let me see if I can find those.
Should be a couple in there for you.
So are there any PK data for Emicizumab? So I think at this point, the question is PK data relevant for Emicizumab? We do have data that demonstrates the metabolism of Emicizumab and so-called pharmacokinetics for the drug, which provided the information to determine the dosing regimen. And we know that we can administer Emicizumab on a weekly, bi-weekly, or monthly basis by adjusting the individual dose. And we don't really understand at this point whether Emicizumab levels in the plasma, how relevant those are to clinical outcomes. I think that's a question that still needs to be addressed. So I hope that answers the data around PK data for Emicizumab. And another question is, how does a patient handle, where to go? How does a patient handle not knowing their factor levels while on Emicizumab? Well, I think that what we know from a population basis is that Emicizumab achieves levels that are relatively standard and provide a degree of hemostatic protection. Again, this is data that's applicable to populations of individuals. And I think that we still have more to learn about the real world experience and real world data on the use of Emicizumab in particular individuals. So I do think that there's more that we need to learn about the individual distribution. And I think if you look at the data from the Cohen paper, as well as the Warren paper that I showed you, there's still people who are experiencing breakthrough bleeding while on Emicizumab. So it's important that we continue to generate data around Emicizumab as well. So are there any other questions here?
Len, there's one question in the chat that I sent you there at the end.
Yeah, so if there's no other questions, I would like to thank all of our attendees, and I'd like to thank Bayer for providing some information for us that we were able to share in here. And I think really some of the insights that I shared with you around the goals and desires for people living with hemophilia were generated through some Bayer-funded studies. And I think it's important that our industry partners continue to collaborate with the community on generating this data so that we can all improve the outcomes for people with hemophilia. So I'd like to thank everybody for participating today. And I'll make the comment for Brett that I believe by Friday, the recording will be available on our website. So you'll be able to review that recording at a later date. Thank you.